So it's a real pleasure to be here. We've heard some really wonderful talks, and I've learned a lot today. I'm really appreciating the interdisciplinary nature of this forum. My work, our work, is really focused at the molecular level. We're really interested in how Ebola works as a really complex molecular machine, how it invades cells in our bodies to take them over, and how we can learn a lot about this virus and how to control it. Learn about where it comes from, its potential reservoirs, potentially mechanisms of zoonotic transmission, by understanding the series of engagements that the virus needs to undertake in order to successfully infect you or a cell in your body. We're really zoomed in here. The virus is a very simple entity, and it's really evolved to exploit your own cellular machineries to gain a foothold and a purchase in your cells so it can make a lot more baby virus particles. So the question is, how does the virus do that? And what can we do with that information to address the broader questions, the more important questions about therapy and where this virus comes from and where it might go? And obviously, this is only a piece of the much larger puzzle that we've been talking about today. So this is a very famous electron micrograph of an Ebola virus virion by Fred Murphy, but that's not really how we think about the virus. We think about it more like this. So this is a 3D model built by a Russian group. And so I don't know how well you can see that, but we really think about the virus as this incredibly complicated and yet simple molecular machine with moving parts that's got modules. It's almost like built like a rocket ship where there are different stages, and each stage has a job, it does that job, and then it's discarded by the virus, which then moves on to the next stage. And it's really a ballet. It's an orchestrated ballet between virus and cell, and the virus has evolved to execute this flawlessly on most of the time. But it's also susceptible to interruption. One of the strengths that we have as humans in our ability to think and dissect the system is we can find weak points to target. So I'm just going to zoom in a little bit to show you that there are these structures on the surface of the virus. I don't know how well that comes through, but these structures are called the glycoprotein, and their job is to get the virus into the cell. And so I'm going to really spend pretty much most of my time telling you about how this glycoprotein works and how it uses our own cellular proteins to help the virus get in and what we can do about that. So now we're going back to more of a simple cartoon of the virus. You can think of the virus as sort of a lipstick or a stripped wire, or however you want to think about it. But at the core of it is a viral genome. It's the RNA of the virus, which is encapsulated or wrapped in a protein coat called the nucleocapsid. This nucleocapsid is surrounded by another layer of proteins called the matrix. And the whole shebang, this whole assembly, buds out of cells as it's being produced and pinches off a piece of the cellular membrane with it. And so this membrane entirely covers the virus. It's made of a lipid bilayer, just like all the cells in our body that define what's outside the cell and what's inside the cell. The virus is the same way. And the virus actually targets one of its proteins, which is this molecular machine that we're fascinated with, called the glycoprotein, or GP, which forms these trimeric structures, so it looks like a three-bladed fan. So the virus puts this protein in specific spots on the cell surface, and it buds out through those spots. So it's now coated with these glycoprotein molecules. And so we like to think of the virus as having two pieces: the payload and the delivery system, just like a rocket ship. Okay, so this is the payload. This is the stuff that needs to get inside the cell where all the goodies are so you can make lots of new viruses. This is a delivery system. It's the membrane of the virus, and that has the glycoprotein on it. And it's the glycoprotein's job to essentially get the payload into the cytoplasm where the virus needs to go. And this one protein, together with the membrane of the virus, does everything that needs to happen to deliver the virus into the cell. So it's the Swiss Army knife of molecular machines. It has many different jobs. It's extremely dynamic, changes its structure as needed as the virus is entering the cell. Okay, and one of the really nice things about proteins like this and the relative simplicity of viruses as opposed to cells in our own body is that they're modular in nature. So this is another cartoon of Ebola virus. And we don't actually work with Ebola virus in the Bronx. I mean, that would just be a really bad headline in the New York Daily News. Instead, we take advantage of the fact that this protein is modular, and we can use a different virus called vesicular stomatitis virus, which is a virus of cattle. It's not a human pathogen, or at least a major one, It's a great lab animal or lab virus because it grows really well. We can produce it in large amounts. And it too, like Ebola, is an envelope virus, which means it has its own membrane. It has its own glycoprotein, which looks very different from that of Ebola, but functionally it's equivalent. It does the same job, which is get VSV into cells. And so what we can do is essentially replace VSV's glycoprotein with that of Ebola. So essentially what we have is a sheep in wolf's clothing, so to speak. And we can do this a number of different ways without getting to a lot of the gory details. A very simple thing we can do is, this is a genome of VSV. So it's got these different genes, and the G gene is the glycoprotein of VSV. So we can do two things. We can put a GFP gene into this viral genome. So we can reprogram the virus so that when it infects a cell, it makes this protein called green fluorescent protein, which is exactly what the name says. It's a green fluorescent protein. So it's really easy to follow infection by looking for cells turning fluorescent green. But the more important thing is that we just replace the glycoprotein of VSV with that of Ebola, and now we have a new virus that doesn't exist in nature, won't kill people, is in fact is actually a vaccine vector. It's one of the leading vaccine candidates. That if you give it to a monkey, it actually protects it from Ebola because of the immunogenic properties of the Ebola glycoprotein. And this virus grows in the lab. So what we've done is we've taken a dish of cells, which you can't really see except for where it's green, and we started an infection at the single point right here with a single virus particle. And that particle is spread radially throughout the culture by going into a cell, infecting it, producing lots of new viruses, which then go on to infect the next ring of cells, and then kill the cell that was originally infected. And so what you're looking at is all these viruses, all these cells are dead, and this is an active ring of viral replication where all these cells are still, are all heavily infected. And you can start to see like faint infection where the infection is continuing to spread. So this gives us a really nice system in the laboratory to understand how this virus works, and more specifically to really understand how the Ebola glycoprotein helps to get the virus inside the cell. And we can do this without putting ourselves at risk and other people around us. And when we find something interesting, I have a buddy who works at USAMRIID in Fort Detrick, his name is John Dye, who actually wears the moon suits and works with live virus, and I call him up and say, hey John, I got a cool result, you want to check it for me? And he said, oh sure man. So more or less like that. And so, you know, that's kind of, we have to confirm everything that we do with the system using the real Ebola to make sure it holds up. And the good news is that, you know, pretty much, you know, I would say that most things that we find in our system hold up with the real virus. And one of the great things about it is that we can move a lot faster working in the Bronx than people can in Fort Detrick wearing moon suits. So this actually makes the research move. So what are we doing with these kinds of systems? So we and other people have been using these kinds of what we call surrogate approaches to study the Ebola invasion process for about the last 15 years or so. And here are some of the things we're learning, okay? And this is a sort of a rough outline. I'm leaving out a lot of details. So we know that the virus has to stick to the cell. I mean, that needs to happen so the virus can get into the cell. But we actually haven't found any specific proteins or any molecules on the surface of the cell that the virus absolutely has to have. Instead, the virus seems to masquerade as a bit of dead cell. So there's this normal housekeeping function in your body that when a cell dies, it gets packaged up into little nice pieces, and the neighboring cells eat it. And then they digest everything, and the cell is like it was never there because it's been digested by all the neighboring cells. And these dead fragments have signals on them that are eat me signals that tell neighboring cells to essentially take them up, okay? And this virus has evolved to put the same eat me signals on its surface. So this cell thinks it's a piece of debris that needs to be digested, and so it binds it and it takes it up, and it takes it into this membrane-bound compartment called an endosome. And eventually, this endosome is sort of like a mining car that has ore that's going to go into the smelter. I mean, essentially it rides on rails in the cell to a compartment called the lysosome that's just the worst place you want to be. It's like one of the rings of hell in Dante. It's got horrible enzymes and, you know, it's like pH 4. It's going to absolutely destroy the virus. And so you normally think that viruses want to get the hell out of there before they get to that point, and actually most viruses do. So for example, flu has evolved to detect the dropping pH or the increasing acidity of these compartments. So when it gets into an endosome, it uses its glycoprotein to blow this endosome and then escape into the cytoplasm, which is where it needs to go. So one of the surprises we got with Ebola early on was that Ebola does not actually do this. Instead, you know, it actually uses these same kind of enzymes that are in this compartment that are used to break proteins down and dismantle them, it actually exploits them in a very specific way to gain its own access. Okay, so, and I'll show you that in a couple of slides, but these enzymes are called cysteine proteases. It's the nomenclature thing that has to do with how the enzymes work. Exactly why they're called cysteine proteases isn't crucial, but we can talk about it later if you're interested. But the point is that the virus needs these enzymes, and these are our enzymes that are in all cells in our body, and the virus has evolved to exploit them. So, so that's sort of one point of vulnerability for the virus. The second is that the virus has evolved to use a protein called NPC1 or Niemann-Pick-C1, which I'll tell you a little bit more about, as a receptor, which means it's a protein that the virus grabs a hold of, and it essentially is part of the virus's GPS. It tells it where it is in the cell and tells it what to do next. So, and it does this by way of changes in the structure of the glycoprotein of the virus. And so the virus absolutely needs this protein, and it needs to bind to it. Okay? And after this binding happens, there are a series of changes that happen which we haven't really understood yet, that essentially result in the glycoprotein of Ebola fusing the membrane of the virus with the membrane of this endosome. So this is sort of like two soap bubbles coming together, but it's catalyzed by this viral protein, and this soap bubble formation process that essentially deposits the payload of the virus, the guts of it, into the cytoplasm, which is this stuff here, that's not empty but is loaded with stuff, that proteins and other macromolecular machinery that the virus needs, nutrients that it needs to actually take over the cell to make millions of little viruses. So that's the sort of end game of this invasion process, this fusion reaction that needs to happen. And so this is actually a little video that was made by my colleague Erica Sapphire at Scripps. And so what this shows is essentially what happens during this fusion reaction. So this is the glycoprotein, it's just a different view of it, where you can sort of see that it's all stretched out like this, and it's in the viral membrane. And it gets cleaved by these enzymes called cysteine proteases, and then that sets the stage for the receptor to bind, and I'll show you this a little bit more in detail. And then you get this reorganization. A piece of it harpoons out and hits the cell membrane. And so then the virus has sort of like stuck itself to the membrane of the cell. And so now we're looking at two of them because this kind of has to happen in a cooperative way. And so the next step will actually pull the membranes together by jackknifing this protein in half, as you'll see in a second. Okay, so this is just a simulation, but it illustrates what needs to happen. This glycoprotein of Ebola needs to undergo these massive series of conformational changes, essentially changing in structure in order to pull the two membranes together and coax them to fuse. This is what this virus needs to do. This is what Lassa needs to do. This is what flu needs to do. This is what HIV needs to do. And any virus with an envelope has to do this. But one key difference among all these different viruses is they use different signals to orient themselves in the cell, to tell them when to do what. Because one of the problems with this fusion reaction is that the virus has one shot. Okay, if you're a virus particle, you have one shot to get it right. If you fuse in the wrong place, if you fuse to a piece of dirt, or you fuse to the side on the floor, or you enter the wrong kind of cell in the body, or you infect the wrong kind of animal, and you fuse, then you're dead. You are selected, you're out of the gene pool, because you had one chance to get it right and you didn't do it. So there are incredible controls through evolution, through positive selection, for these functions to be maintained. And these very things that make these viruses so good at finding the right kind of cells to infect, and so efficiently infect them, also makes them vulnerable, because there are these specific lodestars that they use to tell them what they need to do next. Okay? And so I'm not going to talk much about ZMapp, but ZMapp is a treatment which is really a mixture of antibodies that works by taking advantage of one of the Achilles heels of the Ebola glycoprotein, which is its dynamic nature. So this is a cartoon of a monoclonal antibody, of an antibody from a person. And this is actually a structure, again, from my colleague Erica Sapphire, who is a structural biologist who's done a lot of work on Ebola, looking at what happens when the three different antibodies in ZMapp bind to the Ebola glycoprotein. This is just like a sort of a more blobby version of the glycoprotein. But essentially what happens is like the Ebola glycoprotein is a ballet dancer, and these three antibodies are like essentially like wrestlers that pin it down and keep the different parts of it from moving apart from each other. And that's essentially kills the virus, because it needs to change in structure as the virus is infecting the cell. And so these molecular staples keep it from doing that. So this is one kind of quite effective strategy to treat a virus like this. But I'm going to talk to you about something a little bit different, which is instead of trying to go directly at the virus, can we go after these host proteins that the virus has evolved to use? Okay, so the first host protein that I mentioned is this protein called endosomal cysteine protease, which is really an enzyme that has this cleft where pieces of protein get stuck and then they get clipped. Okay, so it's a molecular scissors for taking a piece of protein and cutting into two pieces. Okay, and so what the virus has done is it's evolved to use this protease, the cellular scissors, to chop off a piece. Okay, and it just seems very subtle, but what's happening is this big chunk of protein is just being clipped off and it floats free. And what that does is actually creates the binding site for the receptor protein. So the virus is able to put the shield on its surface that prevents it from probably being killed by antibodies. But once it's inside an endosome where there aren't any antibodies, it's safe from the immune system, and then it's evolved to come along and be clipped so that the shield can float off because it's not needed anymore. And now you expose the binding site for this protein that needs to bind. So one strategy is to prevent the enzymes in the cell from clipping the glycoprotein and creating this binding site for the receptor. And so any molecule, small molecule inhibitors that target these enzymes should be antiviral, and that's what we've found. So essentially this is a collaboration with a number of different groups to identify new kinds of protease inhibitors that could be effective against Ebola. We've only gotten into cells. We're doing a mouse experiment this week, but these are the experiments in cell culture with real Ebola, not the VSV toy that we play with in our lab, where we're essentially taking these 3 different compounds and treating them, putting them on cells at increasing concentrations, and then we infect them and we monitor the amount of infection. You can see that as you add more and more drug, you're getting really good inhibition, and if you look at the concentration of the drug at which you're blocking infection, these are in the nanomolar range, which is sort of what you want for a very specific drug that's going to go after a virus. So essentially the best compound we have so far has an IC50 of 5 nanomolar, which means half the infection's gone at 5 nanomolar. That's a lot of the other drugs that are sort of, people have talked about that are repurposing FDA-approved drugs, for example, those tend to work in the low micromolar range, so they're 1,000 times worse. Because they're not specifically designed to target Ebola, it's just something that they do on the side. So the other protein I want to talk to you about quickly is this protein called NPC1, or Niemann-Pick C1, which we found along with a group of collaborators a couple of years ago. And this protein is actually a, so it's this protein right here, it's just a cartoon. This is what's called a multi-pass membrane protein because the protein goes across the membrane a bunch of different times. And it's got pieces that hang into the endosome, and that's what the virus is looking for. But this protein is a normal job. It's just like a janitor doing its thing, cleaning for millions of years, and then along comes this virus and says, okay, I'm going to use you to break into the building. So it's kind of like that. So the job of this protein is to essentially get rid of cholesterol from these endosomes as they're taken up by the cell. And when you break this protein, and there is this rare human genetic disorder called Niemann-Pick disease, which is quite horrible because the kids that get it can die pretty young. So they don't have this protein anymore, and so their cells just clog up with cholesterol. But Ebola is using this protein in a slightly different way. It doesn't really care about the cholesterol part. All it wants is a handle to grab on directly to this protein. So it tells it where it is in this particular kind of compartment in the cell. And it actually binds to this very specific part of the protein called domain C. And there's this tight interaction between Ebola glycoprotein and the domain C protein. And that has to happen. And this is just a series of electron micrographs showing you that if you look at wild-type cells, there's normal cells and cells that don't express NPC1, and you kind of watch the virus go in, the virus seems to bind fine, it gets engulfed like a piece of debris, and then it ends up in these endosomes, and all of that looks okay. But what's different in these NPC1 knockout cells that don't have any NPC1 protein is that the virus accumulates in these endosomal compartments. So it never gets out. It can't get out of the endosome, and it's just stuck there, and it eventually gets destroyed by all these nasty enzymes. So essentially the virus needs this receptor protein, NPC1, to blow out of the endosomal compartment, just like flu uses acid pH to do the same thing, but Ebola has evolved to use a specific protein. And so just to show that this is important, we also looked at this in mice, or more specifically my collaborator John Dye in USAMRIID, essentially used mice to ask if this protein is important for infection by the virus. And so this is what happens when you take a normal mouse and infect it with Ebola. Basically, all the mice die within 8 to 10 days post-infection. But if you knock out NPC1, so these mice that don't have NPC1 are basically siblings of the normal mice, okay? So everything else is in common except that they don't have NPC1. And these mice are absolutely resistant to infection, okay? So they don't die. And even more amazingly, the heterozygous mice, which have one normal copy of NPC1 that you think would make them susceptible, they're also substantially protected. So this starts us to think about things like heterozygote advantage in humans, where it's sort of like malaria, where if you had one good copy and one bad copy, you may have some selective advantage against Ebola. Although I have to say this is only in mice, and we don't know if it would apply to humans or non-human primates yet. So this is a bit busy, but really all I want to tell you about this is that if you look at the amount of virus, so in a wild-type mouse, in kidney, you're getting 10 million particles or in 1 milliliter of the sample. Okay, so there's just a ton of virus in these mice. But when you look at the knockout mice, they make absolutely no virus at all at any time. So not only is this protein absolutely essential to kill the mice, it's absolutely essential for the virus to grow in the mice at all. It's sort of like you're injecting mice with saline or something. They don't even notice that it's there. They don't even make antibodies to it. And with the heterozygous mice that have one copy of NPC1, they start off making a lot of virus, so those are the blue squares here. But if you wait long enough, they clear the virus. So they seem to have some kind of kinetic advantage where they are susceptible to the virus, but somehow they're able to have their immune system kick in and kill the virus at a slightly later time. So, so there's sort of this intermediate phenotype, which again is very interesting to think in terms of humans, whether this is applicable or not. So one question is, just like we can with these proteases, can we actually target NPC1 to block Ebola infection? And so to do that, we look specifically for ways to find drugs or small molecules that can actually block the interaction between the Ebola glycoprotein and NPC1. And so we have this fancy assay system that our collaborator at Microbiotics, a small company in Massachusetts, develops, where we can put the Ebola glycoprotein on one kind of bead and put NPC1 on a different kind of bead. And when the beads bind together, and that's really driven by these two proteins interacting like they normally like to, you get a signal because the beads are sort of shoved next to each other. And so you can measure that signal. And then you can also put in different drugs and different compounds. And we're screening 200,000 compounds to see if we can find a few that do this, that actually bind to NPC1 and then block the glycoprotein bead from binding to the NPC1 bead. So if this happens and you know you have a candidate drug molecule that could actually have some utility in terms of blocking infection, we're still in early days with this. We haven't gone into animals yet. But again, in cell culture, just looking at the virus infecting cells, we have 3 different compounds that are all slightly different. So now we're sort of at the point where we're hanging different like ornaments off the Christmas tree to see what matters for the inhibition activity of these drugs. So we have 3 different drugs, and these two, 1 and 2, can block the binding to the virus really well, and they also block infection reasonably well for being first-generation compounds. And this guy, PPZ4, doesn't block binding so well and also doesn't block infection so well. So that makes us happy because it shows us this phenotype of the ability of these compounds to block the virus from binding to the receptor in a test tube kind of mirrors what happens when you do the whole thing inside a cell. And we're hoping that we can go into animals. But again, it takes a really long time for all these things to pan out. And drug companies spend many years getting to really late stage and then find out that it causes dogs to pee funny or something, and then the whole thing's torpedoed. So so we're very cautious, but we're optimistic. So I'm just going to leave you with the last couple of experiments that ask a different question that isn't about therapy, but really about thinking about whether the NPC1 protein could have some implication or some predictive capacity to tell us about what animals can be infected by Ebola and what can't. And this is sort of a tricky problem because we know that Ebola can infect lots of different kinds of animals. We know that Ebola can infect in the lab, lots of different kinds of cells from different species. So our first problem was finding a cell from a species that wasn't infectable by Ebola. And this is a right down evolution lane, but we actually found a snake cell line from this venomous snake called the Russell's viper. So I was born in India and I grew up there till high school, and this is one of the 3 big snakes that are responsible for a lot of deaths. And so I like to tell my dad that I've combined Ebola and venomous snakes, and it's kind of great. So anyway, even though they're both horrible things. So what we did was we took these cells and we infected them with Ebola and also Marburg. And I should point out that everything I've told you today about Ebola is true for every other member of this family. including this strange Yovu virus from Spain. So when we take these cells and we infect them with, this is just a control virus, VSV, which can infect everything, including yeast. So if we infect Ebola and Marburg, that's my time. We need a couple more minutes. We see no infection at all on the by Ebola and Marburg on these snake cells. However, if we take these same cells and we artificially express the human NPC1 protein in them, now all of a sudden they become susceptible. So just putting the human NPC1 protein can take a non-susceptible cell and turn it into a susceptible cell. And this correlates perfectly with binding. Okay, so now we're looking at the binding of the virus to the receptor. This is the human form of the receptor, where the virus can bind fine. And that's the snake form, where the virus can't bind, for really not very much at all. And after a lot of work by one of my students, Esther, she was able to show that, this correlates with infection. So if you normalize human infection to 100%, in these snake cells, you're down by 3 orders of magnitude. So that's a huge effect. Okay? Now, all we have to do is make a single amino acid change in the viper receptor, and we can make it bond. And that gives us normal infection in these snake cells. And all we have to do is make the same mutation in the opposite direction in the human receptor, and that totally kills binding, and it also reduces infection by 99%. So a single amino acid change can have a dramatic effect on the ability of the virus to recognize its receptor. And this amino acid change is a single amino acid, and it's actually even more amazing than that. It's a single hydroxyl group. Just adding or taking off the single hydroxyl group for the chemists out here, it's quite a subtle thing, has an incredible 1,000-fold difference in the ability of the virus to use this protein as a receptor. And by the way, these changes don't really affect the ability of the snake protein or the human protein to do their normal job of moving cholesterol around in the cell. So you could totally envision situations in nature where there's a single amino acid change in some kind of bat that doesn't allow the virus to bind and makes those bats resistant to infection, even though it doesn't really affect the ability of the bat to sort of do its normal cholesterol business. So that's sort of the take home. These modest changes in the receptor interface can dramatically influence host range. Of course, this is all in cells, and we need to go into animals. But I'm just going to leave you with this last experiment that's really looking at whether the Ebola virus and host NPC1 genes could be co-evolving, which kind of expands this question to what's going on there in nature and what could have happened in the past. So this is really based on this notion of the Red Queen hypothesis that was articulated by Leigh Van Valen, an evolutionary biologist, which really is an idea of an arms race, where the virus is constantly mutating and adapting, and so we have to constantly adapt to deal with the virus. We're both constantly in motion, but we're relatively in the same place to each other because this is just like the US and Soviet Union. We keep having more and more nuclear missiles, but during the Cold War, but we didn't really gain a selective advantage over each other. So it's that sort of a process. And one of the characteristics of this kind of coevolutionary arms race is that you see a lot of mutations that are adaptive. You see certain positions in proteins becoming highly diversified, because normally mutations in proteins tend to break them, and so proteins tend to evolve to avoid those kind of changes happening. But at certain places in the protein, you can see a lot of rapid evolution, and that is indicative potentially of these kind of coevolutionary wars. And so this is actually a great example from our collaborator, Sarah Sawyer, who's looking at a human protein called the human transferrin receptor, which is a lot like NPC1, except it's a housekeeping protein for iron, not cholesterol. But it's also a receptor for 2 different viruses. One of them is our South American arenaviruses, which are relatives of Lassa. So Lassa uses a totally different receptor, it doesn't use this protein. There's also positive selection evolution in the Lassa receptor, by the way, it's very interesting. But this protein is actually a receptor for 2 different viruses. One of them is our arenaviruses from South America. And the second is a rodent virus called the mouse mammary tumor virus. And the amazing thing is that you can find these residues in the receptor that are positively selected, meaning that they're rapidly diversifying in 2 different places that correspond to the 2 different spots on this protein where these 2 different viruses bind and use this protein as a receptor. And these spots have nothing whatsoever to do with the normal iron transport function of this receptor. Okay, so basically these viruses are driving the evolution of the transferrin protein in different sets of species, of course, because there's this sort of race going on. And so we want to know if that something like that could be happening in bats. And obviously the receptor is not the only protein you want to look at. You want to look at any protein in the host that interacts directly with the virus. as a potential place where this could happen. So, I mean, Kevin already eloquently talked about these potential bat reservoirs for Ebola. So one thing we did in collaboration with Sarah Sawyer was to take what was available in terms of NPC1 sequences for different kinds of bats, and the colored species there are African frugivorous bats. Plus we sequenced a bunch of things ourselves from samples and cells to try and build a portfolio of bat sequences that we're still continuing to do. And then we actually looked to ask if NPC1 is under positive selection. Are there any residues in this protein that are changing more quickly than you would expect? And we really only find the signature in one place, and that's in the same domain of this protein that's binding to Ebola. So it's early days and we have a lot to do, but I think we feel optimistic that there's something really interesting going on here vis-a-vis Ebola and bats, and potentially other filoviruses. Not even necessarily Ebola, just a filovirus of some kind. Could be something totally different that we haven't even identified yet. So we're optimistic about this lead, and we're thinking that this type of strategy could be helpful to look at other kinds of interactions between the virus and the host as well. So the conclusions, just a summary of what I showed you. The viral invasion mechanism of Ebola and other viruses, they're complex. They make extensive use of host molecules. So these host molecules are antiviral drug targets with potential advantages. I'm not putting down all these other therapies that are out there. ZMapp is actually kind of amazing and incredible that it works as well as it does in monkeys. But there are advantages to these host-directed therapies. So first of all, ZMapp will not work against any other strain of the virus except Ebola Zaire. If you could find a really good molecule to block NPC1, it should protect you against every single filovirus we know about. And I would eat my hat if we find a filovirus that does not use NPC1. I will make a bet with you. The other advantage is drug resistance. So potentially an antibody could engender much more viral drug resistance because the virus can mutate directly to change the binding site where the antibodies can bind. And we're already seeing that with ZMapp in monkeys, not in a way that would be nervous-making about therapy, but just that if you look hard enough, you can find these resistant variants showing up in response to antibody treatment. That's exactly what you'd expect, that problem. So defining the virus-host molecular interface, we think could be really helpful to understand how and why Ebola killed its zoonotic hosts, but perhaps not its natural ones, and maybe give us some clues about barriers to interspecies transmission that would add to the complexity of the picture that includes ecology and all behavior and all these other things, not just of humans, but of the host climate, all of that stuff. And then understanding how this interface has co-evolved in evolutionary time and is continuing to co-evolve potentially could help us learn something about the natural history of these viruses, which remain enigmatic, and may help us to figure out what the reservoirs are, because in principle, one molecular signature of the reservoir could be that specific genes that are in contact with Ebola that Ebola needs would be under accelerated evolution, and perhaps NPC1 is the first example of that. And so with that, I'd just like to thank everyone in my lab. It's not actually everyone in my lab in these pictures. We need to, Anna's supposed to work on the website. And Anna's work is right here from my lab. She's an alumna of this fine institution. So actually a lot of the work that I talked about today was done by two students, Esther Ndungo and Melinda Ng in my lab, and two former students, Tony Wang and Emily Miller. And we have a huge cast of collaborators without whom and all the stuff I told you wouldn't be possible. Particularly shout out to John Dye, who did all of our live virus work, and is really a close collaborator on every project that we do. Okay, with that I'll stop. I'm already late.